Hi, my name is Mia Jones, and I will be presenting on the topic of elucidating the psychological function of the human B12 trafficking protein, CBLD. Before I begin, I would like to give credits to my lab mentors, Dr. Julie, whom I work very closely with, and Dr. Ruma Banerjee. Thank you to UROP for providing me with this phenomenal opportunity. To begin, the Banerjee Lab and I have been researching vitamin B12 defects present within humans. Mutations located in the MMA-DHC gene are responsible for the CBLD defect, which is a protein located in the vitamin B12 trafficking pathway. To better understand the role of CBLD in the pathway, we transfected human cells with expression vectors for epitope tagged wild type of mutant type CBLD. You will hear me refer to these tags as flag tags. The overall goal of this presentation is to study how patient mutations impact the localization and metabolism of B12. Using the generated expression vectors, I established Western blot assays to compare cytoplasmic versus mitochondrial localization of CBLD. I'm sure you all are wondering what vitamin B12 is and what makes it so important. Well, vitamin B12 is an essential nutrient that we obtain through our diet, meaning the body doesn't produce its own. CBLD is one of many proteins within the B12 pathway, but we do not know that mutations in the B12 pathway have been classified into 9 inborn errors. I produce 4 different mutant wild type vectors that are listed on the left. These vectors show the placement of the DNA sequence in which the mutation should occur, which was also checked with DNA sequencing. A flag tag was attached to this vector to be able to identify the location of CBLD within the cells of different cell types with alternate mutations. This next image also explains the process of transfection. 2 different cell lines were used from the colon and kidney for 3 time points of 24, 48, and 72 hours. After incubation, the cells were collected to go through Western blot analysis. Western blot is a rather complex experiment with many tedious steps. This figure demonstrates this process. There are 6 main steps within Western blot. The first step is transfecting the cells, running the protein samples on gels to be visualized by transferring gels to a membrane, blocking the membrane with the milk solution, which just coats them with a protein that will allow the primary and secondary antibodies to stick to the proteins of choice, which are the last 2 steps. After each application of antibodies, the membrane is washed and finally used for detection using a camera that captures the glow that the bands give off when antibodies are present. This is an example of a gel transfer membrane of the kidney cell type transfected with the CBLD flag tag. We use molecular weight markers which assist in identifying the proteins by their weight. Empty vectors are used as negative controls, and CBLC flag tags are used as a positive control because we knew what to expect. The lines that are present on each column are basal CBLD concentrations. However, we would have expected there to be a secondary line similar to columns 5 and 6. The secondary line would ensure that the flag tag is present, resulting in a thicker line of separation on top of the baseline amount of CBLD. From this, we can conclude that the CBLD flag was not detected, although the control CBLC was detected. There are 2 possible reasons why. The flag tag will be unstable when interacting with CBLD, causing us to not be able to visibly see the tag, or the CBLD flag expression was simply too low initially to be able to detect it through Western blot. So to combat this issue, we plan to mutate flag tag to a MYC tag, which we will determine if it's more stable than interacting with CBLD. We will repeat this process using the mix tag, which includes the effect of mutations and investigating the localization of CBLD in the mitochondria and cytoplasm using a mix tag instead. Thank you all for listening to my presentation. I hope you enjoyed and learned a lot.